So yeah, thank you again for the introduction. I need to go and look up the statistics. I didn't know that, so that's good. So when I was asked to give this talk, I was sort of tasked with giving sort of the plant pathologist perspective of this topic. So I'm definitely a fish out of water at this meeting, or a bird out of cage. I'm not sure how you want to put it, but I'm very happy to be here, and thanks everyone for sticking around to the last day for this. So connecting the dots, plant pathology meets poultry biology. So I am in the toxicology and mycotoxin research unit. The unit has been around since the 80s. Initially sort of formed to address the very early days of fumonisin that was occurring. We underwent sort of an identity crisis a few years back, and that was sort of tied to the center, because I'm in the U.S. National Poultry Research Center, which underwent its own sort of rebranding about 10 years ago. To better integrate into the center, we changed our focus, adapted our focus to focus on feed issues, and primarily poultry feed. But we are a diverse group, and we do encompass everything from the plant pathology, the pre-harvest aspects of mycotoxin-producing fungi infecting crops, with the goal of trying to address control at that level. And then, of course, we do focus on feed as well. So if mycotoxins do get into the feed, what sort of strategies, monitoring, and so on can we utilize there to address the issues? And then, of course, the impact on the birds. So all of that is basically input impacts output. Maybe a little more polite way to say, quality in, quality out, instead of garbage in, garbage out. But this is our focus. And these are the people that are primarily involved at each of those stages. So myself and Dr. Scott Gold are the plant pathologists. A lot of our work focuses on biocontrol, which I will get into and talk about. Anthony Poco-Akins, who is here, focuses more on the feed aspect, mitigation strategies, and so on. And then Dr. Shanmugha Sundaram, who will be speaking later, addresses more of the actual direct impacts of these mycotoxins on poultry. So, and then I do want to point out a support scientist, Dr. Tim Satterley, very talented fungal geneticist that has also been integrating with Anthony and working on some of the feed aspects as well. So I think that's all I wanted to say here. This is just to drive home the same point you all know. You've been here, you've heard the previous talks, you know what's going on. Basically, mycotoxins, when we talk about corn, I refer to corn as it's a condo for fungi, because there are lots of fungi will infect corn, oftentimes not causing any problems, but when they do, especially in terms of mycotoxins, it becomes a real issue. So corn is very susceptible to infection by a lot of these different mycotoxin-producing fungi, and of course, corn is a primary feed ingredient, so nothing new there. These are the primary mycotoxins listed across the top here, and it's already been kind of pointed out. I think it was the quote from Mike that about fungi having tremendous impact on plants and animals. When it comes to mycotoxins, there's really two genera, Aspergillus and Fusarium. Penicillium sometimes, but Aspergillus and Fusarium species are the dominant ones. And in terms of deoxynivalenol, primary Fusarium graminearum, for fumonisin, primarily Fusarium verticillioides, and then for aflatoxin, primarily Aspergillus flavus. These values shown in the table are either guidance levels, whether that's regulatory or just advisory. But the thing to keep in mind is these levels are set one toxin at a time. And a lot of these values may need to be reevaluated, especially with the growing understanding that we're dealing with multiple mycotoxins at a time in terms of infecting corn. And this is a paper from Dr. Shanmugha Sundaram's work, which she may touch on today. But just to point out, so if the regulatory levels for fumonisin and DON in poultry, I'm sorry, I'm having a little hard time seeing from this angle, but basically for fumonisin, 20 parts per million, and for DON, 5. In her study, she looked at very low levels, so subclinical doses of these two toxins combined, and showed that it was still having noticeable impacts on poultry. And and in this case, predisposing them to Clostridium issues. So even at subclinical, subchronic doses, the combination of these toxins is still likely having a significant effect. In terms of control, when it comes to trying to control these fungi, fungicides just don't work, especially when we're talking about corn. Corn is such a large plant, it's so many acres. The sheer volume of fungicide that would have to be applied to even be efficacious it's prohibitive. So one technique that has worked well for aflatoxin is the use of atoxigenic strains, or non-aflatoxin producing strains, to be more accurate. And the basic idea is here is you can easily, I say easily, fairly easily, screen different isolates of Aspergillus flavus, and chances are you're going to be able to identify several isolates that don't produce any aflatoxin. This is done in vitro using molecular methods as well as analytical chemistry to identify these strains that don't produce any aflatoxin. You can take those strains and then artificially inoculate a field with them, and it will displace through some sort of competitive exclusion or competition, what have you. The atoxigenic strains will displace the toxigenic strains, and you'll get pretty effective control. Sort of 80 to roughly 90% reduction in aflatoxin levels using that product. Great, works great for aflatoxin. For fumonisin, for fusarium in general, not so much. The biology of these fungi are very different. The way they interact with the plant is very different. And the idea of utilizing a non-toxigenic Fusarium verticillioides isn't really going to work, primarily because of one reason: it's really hard to find them. And that's a major distinction also to point out between Aspergillus flavus and Fusarium species. Flavus, you'll often find atoxigenic strains. In Fusarium, very rarely. So there's something about, in this case, fumonisin, that this fungus, even though it's very costly to produce this compound in terms of energy and so on, it's still doing something that, to the point that the fungus just doesn't want to get rid of it. One thing that does work is transgenic corn, so BT corn, because this often results or is triggered by insects feeding on the ears, on the kernels. And when that happens, carbohydrates are liberated, the fungus can go crazy and start to colonize and cause this ear rot. But if you can prevent that insect damage by using BT corn, then you can dramatically lower both the amount of ear rot and the amount of fumonisin contamination in the corn. But what other tools could we possibly utilize? And sticking with the idea of biocontrol, we've been doing a lot of work with this fungus here called Serracladium Z, used to be known as Acremonium Z. Mycology is dear to my heart, but the name changes do get annoying. But this is now known as Serracladium Z. And you can see from the pictures the effect, the antibiosis it's having. This is flavus here. So the smaller culture is Serracladium. This is Aspergillus, and you can see the effect it's having there. This was just a contaminant, but I love the picture. This is Cladosporium. It's floating around in the air right now, landed on the auger, and you can see it is trying to literally lift itself up off the auger to get away from Serracladium. It also kind of looks like a little alligator with its eyes sticking up. But this is a good example of the antibiosis that this fungus is capable of. So we've been investigating this a lot, and so for the remainder of this, I want to go through this sort of topic to show you some of the data that we've generated. One of our grad students, Nino, picked up on we had done a preliminary survey in Georgia to determine what fungi we were finding on corn, feed corn, grown in Georgia. And in 2019, we did the study. It was actually a little side story. My son had where's my wife? Was it junior high or freshman year or something? He had a project, and we were like, hey, come on into the lab. And so we had him he and his friend do some preliminary screening. He never came back to the lab after that, but I guess he got his fill. But anyway, so Nino picked up on this and did another survey, a more extensive survey, but the results are basically the same. Half of the fungi we're finding off of, and again, this is asymptomatic corn kernels. So no year rot, healthy looking kernels, surface sterilized and plated out. Half of what we were finding was Fusarium, and great majority of that is verticilioides. Surprisingly, or at least initially surprisingly, but I think Alexandra's data kind of, or something she said may have touched on this as well, but we found very little Aspergillus. And again, it's asymptomatic corn. Aspergillus tends to be more associated with some damage and storage. We found this penicillium. Don't really know what to think of that yet, so I'm just going to skip over it. But then the other, we found about 10% of the isolates were the Serracladium Z. So we are looking at these more closely, or Nino is, and trying to identify potentially good biocontrol strains. So, and just sort of an example of some of the extremes that he found. This was kind of interesting. In this county here, Bartow County, one of the fields, it was pretty much all Fusarium. Everything that was isolated there was pretty much all Fusarium. But then down in this county in Macon, one of the fields, it was nearly all Serracladium. And that we're kind of excited about. So we're going to try to follow up on this observation. But that's just an example of some of the extremes that he found. So these two fungi, Serracladium Z is an endophyte of corn. And endophyte, if it's not a term you're familiar with, basically it's just refers to microbes, bacteria or fungi, that can infect plants for some or all of their life cycle and typically don't cause symptoms. Unless some sort of trigger causes that. So these fungi are endophytic. They grow within the plant themselves. And on this, I don't know if you can see from the audience, but anyway, there's some tiny little colonies that are Serracladium, and then there's this major colony here, which is verticilioides. Serracladium is much slower growing than Fusarium. But we found that these fungi interact through their metabolites quite extensively. And to walk you through this model, because this is going to come up a couple of times. Going all the way back to the 60s, it was noted that if you go in and dissect kernels, you will get, or you can commonly find these two fungi in kernels. But if you start to dissect them out in terms of their parts, the parts of the kernel, the embryo is often where you would find Serracladium, the endosperm, or the pedicel, the tip of the kernel, is often where you would find Fusarium. Not all the time, but that's a good sort of general rule. And we think part of that reason is there's sort of a chemical interaction going on between these two fungi. If they are both infecting this very small kernel, a relatively small real estate, one confined to one portion of the kernel, the other confined to a separate portion, we think it's due in large part to these porosities. These are metabolites produced by Serracladium. They were shown back in the 2000s to have some antimicrobial, antifungal effect. By another ARIS researcher who has retired. And so we picked up on that and following up on this porosidine story. Let's see. But anyway, regardless, through this interaction, there is an antagonism that's going on between these two different fungi, and these compounds play a role in that, as well as others. Okay, yeah, so, sorry. The two porosidines that are produced by Serracladium, porosidine A and porosidine B, not going to get into much into the chemistry here. Just a main point is that they differ only by a double bond in this lactammarine. So that's the only two differences. But porosidine A has been noted to be more biologically active, and that's also what we found here. This is some work from another student, Lily Lofton, looking at the growth. This is growth curves of Fusarium verticilioides wild type that has been, these two here are the controls, so you can see they're growing fine. This is with porosidine B only, this is with porosidine A only, and then when you combine them, the fungus is not happy, not able to grow. So that was exciting and gave us some data to work off of. This was also a very exciting result. If you take the porosidines and you dose Fusarium verticilioides with these, at levels that are not toxic, so basically below 10 micrograms per mil. I guess I should have pointed out, this was all done at 20 micrograms per mil. But if you go to sub-inhibitory doses and start dosing fusarium with porosidine, in this case it's just porosidine B in this particular graph. So these are the different doses along the bottom, and this is the concentration of fumonisin in those cultures, looking at the individual fumonisin isomers, FB1, FB2, and FB3. If you get down to 2.5 to 5 micrograms per mil, porosidine will basically cause the fungus to stop producing fumonisins, which is really exciting and was unexpected when we first looked at this. So to go back to here to the model, we do believe that porosidines in combination are having a direct impact on fusarium and its production of fumonisins. A former grad student of ours, Dr. Minglu Gao, who's now with Syngenta, he dove in a bit deeper on this whole topic by looking at some RNA-seq experiments. Basically, we dosed the fusarium, the fumonisin-producing fusarium, with the porosidine A or B at these sub-inhibitory levels. And I'm not going to go through the whole table, just a couple of things to point out. There was an ABC transporter that was, these are log2 fold change. So around 6 to 8 log2 upregulation of these ABC transporter. And I'll talk more about that in a second. But the main thing I wanted to point out was this gene here, ZBD1, log2 of 12 upregulation. That's over 4,000 fold. This gene comes on hot in response to porosidines. We looked at the other interesting aspect about ZBD1, good real estate. It's sitting right next door to the fumonisin biosynthetic gene cluster. Gene clusters, if you're not familiar with them, secondary metabolites in fungi are made through these gene clusters. They're clusters of genes that are co-regulated for a single purpose, which is the production of secondary metabolites. This gene that we saw turn on at over 4,000 fold in response to porosidines is right next door to the fumonisin cluster. And again, when that gene came on, we're dosing it with porosidines, fumonisin production dropped out. But what we didn't expect is when we knocked out this gene in verticilioides, and that's easily done with these fungi. They're haploid. You can do haploid genetics on them pretty easily. To orient you here, so again, we're looking at FB1, FB2, FB3. The very small bar at the far left, that's the control. That's the amount of fumonisin being produced by verticilioides under normal conditions. When we knocked out this ZBD1, fumonisin production jumped up over 40-fold, which was weird and exciting. And then when you add the gene back in, it goes back to being wild-type levels of fumonisin. So what does all this mean? So here again, coming back to the model. So again, we're talking about interactions in a very small bit of real estate. Sericladium is producing these porosidines that are impacting fusarium. And so inside the fusarium cell, these porosidines are detected. There's an ABC transporter that's needed to transport them out of the cell. It's a defensive mechanism. These porosidines also turn on, to a very high degree, this ZBD1 gene, which I didn't mention before, but prior to this work, we didn't even know what this gene did. It didn't really show much gene expression changes. Of all the previous papers that happened to be looking at it, it really wasn't clear what it was doing. So this was the first bit of evidence to suggest that it's having a direct involvement in this interaction. But again, so the porosidines cause this gene to be upregulated, which suppresses the fumonisin cluster. So when the gene's on, fumonisin isn't being produced. You take the gene out of the equation by deletion, and it de-represses fumonisin production, and it shoots through the roof. So a lot of interesting biology here, and we're still trying to tease it all apart. But it all looks promising in the sense of leading to a possible biocontrol strategy. This is Tim's work. Just to quickly go through this, I've been talking a lot about verticilliorides, Fusarium verticilliorides, but I just want to show you that this fungus also is having an impact on Aspergillus flavus and aflatoxin production as well. It is a slow grower, so we have to, in these assays, we get better results if we give it a head start. So this is the Serracladium, and this is Fusarium verticilliorides, and this is Aspergillus flavus. At 12, if we give the Serracladium a 12 days head start, you can see its efficacy is much better than, say, at a 3-day head start. And this is just to show you that in these zones of interaction here, if we look at the actual aflatoxin production levels in those, these are the controls, and then we can see that the Serracladium is reducing the amount of aflatoxin being produced. But what we can say, this reduction in aflatoxin, what we can say at this point is that it's not due to peroxidines. And I know I'm throwing a lot of different metabolites at you and different interactions, and so I'm hoping I'm not losing you. But what we have also done is we've identified the genes in Serracladium that are necessary, the gene cluster that's necessary for the production of peroxidines. We've knocked out one of those key genes, so now Serracladium can't produce peroxidines. That mutant is still being very effective against, that's the mutant here of Serracladium. It's still being very effective against Aspergillus flavus. And these are just two different strains of flavus that have different morphologies. So we see an antibiosis going on against flavus. We see it's knocking down aflatoxin production, but in this interaction, it's not due to peroxidine. It's apparently due to something else, which we haven't identified yet. And this is just to show there's another species of Aspergillus parasiticus that also produces aflatoxin, and the main point here is just to show that it's having the same effect on that species as well. And then in terms of applicability, what we're thinking is that Serracladium could be utilized as a seed treatment because like I say, it already has an association with maize. So if we can apply the Serracladium as a seed treatment, and that's what this was attempting to mimic. So here's the controls kernels here. This is with Aspergillus flavus only growing on the kernels. And then this is with flavus and Serracladium growing on the kernels. And you can see there's very little flavus growing on the kernels. And then this is the assessment of aflatoxin production. And you can see that there's very little aflatoxin there. Fungi beating up on each other. And just to, again, address the issue of variability, these are 3 different strains of Serracladium. You can see there's a lot of variation in in the morphology of the species. Also in its secondary metabolic profile. So this particular strain is very effective at knocking back the Aspergillus that's plated on either side of it. And this is the same Aspergillus on these others. You see this strain is definitely knocking it back more. That's just unfortunate contamination on that plate. So take-home message, we're targeting this fungus, Serracladium Z, as a possible biocontrol. There's still a lot we need to learn about the fungus, but for today, I just want to point out that a couple of things that we need to take a look at relative to poultry is we need to do toxicity studies with Serracladium Z and the peroxidines to make sure that we're not swapping or adding problems. But from what we've seen so far, we don't anticipate that being an issue. But still, that's something that needs to be done. And then we need to take this to the field. Everything so far I've shown you has been in the lab, but it seems to be very promising, and so that's the next step there. And I believe that's it. So we adopted the Phoenix chicken as our logo. I don't know if you noticed that in the beginning with the, but it was sort of symbolic of our unit's rebirth into a different focus on poultry. So anyway, thank you very much, and I appreciate the opportunity.